Um, Good evening, participants. Welcome to the sixth session of certificate course on artificial intelligence and machine learning approaches of drug and vaccine design in cancer. I think I have shared you the sixth presentation related to molecular talking analysis and virtual screening. So before going to this chapter 6 on these topics, okay, can we recap what we discussed in the last session? So what are the last session topics? So can you discuss about the last session topics? What exactly did we discuss in the last session? What is the major tool? Yes, good. We have gone for molecular talking demo using Autodoc. So I can ask you a few questions in the Autodoc. So what is the algorithm we have used on yesterday in Autodoc? Actually, we have used algorithm for running the Autodoc, isn't it? So what is the algorithm we have used for running doc in Autodoc? Actually, we have followed majorly two important procedures. One is running grid in Autodoc. The other step is running doc in Autodoc. So what is the algorithm we have used? It's just a recap, and if you find the same, very good. The American genetic algorithm is the correct answer. Good. Yes. So then I can go for the second question. Uh, so the second question is about what hydrogens we have added. We have added in initial step of Autodoc. There are types of hydrogens, isn't it? So what kind of hydrogens did we add in the initial step of Autodoc? We have added charges. We have added hydrogens. What hydrogens and what charges? Also mention me about charges. So we should remember each and every step in the Autodoc. So like if you complete any docking in Autodoc and if you try to publish in journal papers, you have to mention all this. Non-polar hydrogens? No, that's not the exact answer. All hydrogens is right. Good. The answer is all hydrogen. And how about charges? What charges did we add in Autodoc? What is the name of charges? They have types of charges, isn't it? We have used gas Steger charges. Okay. We have added gas Steger charges. Like, we have positive and negative charges. Only we have positive and negative. Of course, we also have neutral. But how much positive, how much negative? So here we are using this kind of gas Steger kind of a charges. And the next question is related to what is the next software? What is the software we used for visualizing 2D and 3D interactions? We have used a visualizer software for viewing 2D and 3D interaction of ligand receptor complexes. Yeah, what is the software for viewing 2D and 3D structures of ligand receptor complexes? Yes, sir. You can unmute and answer if can't type in the chat box. Very good. Discovery, but is that a software? Discovery is a software. Biovia is the name of the company. Biovia is the name of the company still. What is the name of the software? And in that package, you also have one module, which is important, not Autodoc protocol. I have sent Autodoc protocol just now to your coordinator. He will be circulating it soon. I'm asking you, what is the visualizer we have used? In my protocol, I have showed like PyMol, but I have committed on, but I have committed on um, a tool. What is the name of the tool? No, not Doc UCSF. It is BioVia Discovery Studio Visualizer. Okay, in BioVia Discovery Studio, we have plenty of modules. If you want to dock a ligand with a receptor, you can use DS ligand fit. If you want to uh, view the scores after the docking, you can use DS ligand score. And if you want to go for QSAR analysis, we also have a software called QSAR, DS QSAR. Likewise, we have plenty of modules in a single package called Discovery Studio. BioVia, of course, is the name of the company, the supplier of the product. Now, I think I can go ahead on today's session related to how to go for molecular docking analysis. And how about virtual screening methods? Before going to this, any other thing we have discussed other than the Autodoc, DS visualizer? We have discussed about theory in docking, isn't it? We can also recap about that. What are the three theories we have discussed? Three theories discussed in docking, in formulating a docking software. Okay, so if you see the theories, we have seen, yeah, if you can able to answer, you can unmute and answer, or you can type the answer in the chat box. What are the three theories? Number one, can I give you a clue? Good. Lock and key. Good, Sahana. So actually, lock and key by Fisher. Good. I got all the three answers from a single person. Praveen. Induced fit, conformation theory, and lock and key. Actually, if you see in hierarchy, initially we got lock and key, then we got induced fit implemented in the software, then conformational theory. If conformational ensemble is the correct word for that. Good. My next question is, what docking is not possible based on rigidity in ligand and receptor molecules? I told you there are three possibilities of docking a ligand and receptor based on rigidity and flexibility. Good, Sangami. But my question is, only one possible, there is, for one docking, we can't able to make it with the ligand and receptor. What is that? Making protein rigid and ligand flexible? No, that is not the correct answer. Flexible? No. Ligand being receptor, no, I can't understand. Ligand rigid, okay, and a receptor is flexible. Very good. Is that right? No, you totally confused me. you, uh, actually, you are telling ligand is rigid. Is it? Ligand is rigid and protein is flexible. Okay. I can take you to the PPT. So the one which is not possible is having a rigid ligand and flexible protein. Rigid ligand and flexible protein alone is not possible. Okay. So at last, a combination of answers from two different participants is the right answer. Totally confusing. No, actually, whatever answer is right initially, the Jayanep Jaffer and Sania Kalandar both have answered. I am notified, but for confirmation, okay, I just showed you. Okay, so like keeping the protein flexible and having a ligand rigid, this docking alone is not possible. So you can't find any softwares based on these principles. Okay, there is no confusing. You simply remember like that. but remaining combinations are possible. And if you see, if you have both the ligand and receptor as flexible, you have to remember the induced fit. Okay, if the ligand is also having a conformation, induce, you can find it can have a conformational change, and even if a protein is having a conformational change, like a ligand is molded to fit to a protein, obviously this kind of software, the docking software construction is difficult. Okay, programming is difficult. Okay, this is something about the flexibility of ligand and receptor. Let me go ahead with the new chapter called molecular docking analysis and virtual screening. So, virtual screening, I'm going to take you to Autodoc PYRX. Okay, the literature paper, what we are going to see as a case study is whatever we seen on yesterday. In 2022, a paper published on computational studies of a plant compound beta-ferine. This is a plant compound steroidolactone from ashwagandha, can be used for treating cardiovascular diseases. Okay, so I just targeted two important receptors. Initially, I've taken multiple receptors, but these are most determining, most important receptors we got as a target, and our plant compound was docked well with this kind of receptors. If you see this kind of a results, okay, initially I've shown about the autodock energy. So initially I just got with, if you see the receptors, I've taken totally 9 important receptors responsible for cardiovascular diseases. These are the markers. Okay, from these markers, if you see the ligand, whatever I mentioned as a ligand, is the drug used in market, okay, to target the kind of marker proteins. Okay, so the drugs generally used to block this, or the control drug for PAFA is MAFP, and for this receptor rucaparib, and likewise. So whatever we have, okay, these the 9 receptors, we have these as the 9 control drugs. But in my study, I tried with a plant compound with a friend A. So what I did is whatever uh, plant compound I have chosen for docking with the 9 protein receptors, I've also checked with the control drugs, and I just compared which is best. In addition to the interaction energy from the autodock, I also gone for estimation of DSX score, which is a summation of a drug likeness scores. Okay, so it's like a drug likeness scores. I've just compared both the ligand and vitaferin A. So the interaction energies I got for each and every receptor, I just compared the control drug with the vitaferin A, and similarly DSX scores. And also in the second table, I just I just drawn a data, okay, with the atoms from the compounds, an atom from protein targets. I also found the length of bonds, okay, and atoms from WFA, atoms from protein targets, and length of bonds. So how to analyze these kind of results? We didn't discuss in detail on yesterday. Since today we'll be discussing how we can get these kind of a data after talking. And then similarly, in 2023, I also published a paper with a compound where we talked with the two important receptors of hepatocellular carcinoma. Here also, I've used the similar kind of a table. I used a ligand, the binding energy of my plant compound and a binding energy of a control drug for hepatocellular carcinoma for second and third stage, that is sorafenib. I just compared with each other. So if you see, my plant compound is showing negative maximum energy with most of the receptors. I think in almost all the receptors, we got negative maximum energy when compared to the standard control drug sorafenib. You remember on yesterday I mentioned you, if you try to dock a ligand with a receptor in autodock, whatever energy you are going to generate, these are generated in units, kilocalorie per mole units. Okay, and the significant energy should be -6 and below. Okay, so almost if you see the whatever I talked with the Bergnon B with the 5 receptors, I got the significant results, except for sorafenib, I didn't get significant result for BCL2 and caspase 3. Okay, so also if you check So that means that the sorafenib is targeting majorly VEGFR, EGFR. Even if you check it in your literature papers or even drug databases, it's clearly mentioned this is a standard control drug for these two inhibitors, VEGFR, EGFR. Okay, but it's not well with the apoptosis kind of a receptor, caspase 3. But of course, it shows something well for a caspase 9. Okay, let me see what are the other data mentioned in the analysis. So there is a chain of a protein atom. Okay, so the what chain I've used? A chain. An atom, amino acid atom is a glutamic acid. Position of the glutamic acid is 257. And what is the atom position of my plant compound? And how about the atom position in this glutamic acid 257? I got two different positions. That means my plant compound pyrimidine B at O6 position is interacting with two hydrogen bonds. One hydrogen bond is shared with O and another hydrogen bond is shared with OE3. And similarly, if you estimate the bond length I got is 2.6 for first hydrogen bond and 2.56 for the second hydrogen bond. So similarly, even it was calculated for the other receptors. Okay, so the just hydrogen bond information alone is shown here. But in 2D interaction, do you remember yesterday I showed you something related to pi alkyl bonds, some conventional hydrogen bonds. Okay, so many kinds of bonds. I've shown all these things in the 2D interactions. Okay, so of course that is also a kind of a data. Okay, in 2D only you will be getting all the covalent and non-covalent bonded interactions of ligand with the receptor. But here in this table, you can show the hydrogen bond interactions. And if you see we have one more article published in 2020 in Elsevier journal.
Sorry, sir.
Yes.
Sorry to interrupt, sir. Could you show the table once again?
Yeah, you can see the table, sir.
Could you explain it once again? The bond interactions and everything.
Yeah, actually, if you see the bond interactions, pyrimidine B is the desired drug. Okay. So if you see the docking of Berivnon B with VEGFR receptor, that is vasculoendothelial growth factor receptor, the energy what I generated after docking is minus 8.04 kilocalorie per mole. And if you see my atom of O6 position in Berivnon B is interacting with A chain of glutamic acid 257 in these atom positions. One hydrogen bond is shared with O of this glutamic acid 257, and another hydrogen bond is shared with OE3 of glutamic acid 257 of chain A. Okay, and if you see these hydrogen bonds length, okay, the length for O6 to O is 2.6 angstrom, and O6 to OE3 is 2.56 angstrom. You got it, sir? Now you got it?
Yeah, sir. Yeah. Thank you.
Good. So likewise, okay, even you can check you have to fetch the data for even the other protein receptors after the docking process. And this is the third article I am showing it that is related to in silico studies reveals immunological signaling pathways that genes and potential helpful targets in ovarian cancer. Do you remember in the previous session, the previous class, I mentioned you something that in the methods of drug design, computative drug design, I have used a kind of system biology approach, system pharmacology approach. And later, after system pharmacology approach, I just finalized top 10 genes that which are responsible for ovarian cancer. The top 10 genes are from both upregulating and downregulating stream. Okay, I'm not bothered whether it's coming under upregulating or downregulating, but these 10 genes, whatever I'm going to filter at final, are the most important target genes responsible for ovarian cancer. So I have gone steps of procedure to finalize the 10 important genes. Later, I just talk with three important plant compounds. Okay, so this is the article. It reveals what is the best plant compound talked. If you see the three plant compounds, what I use, all the three plant compounds I have drawn from plant sources, quercetin, epigenin, resveratrol. All these three are plant compounds. If you see this kind of an information, okay, I told you, of course, whatever internal binding energy we generate from Autodock or most of the docking softwares, that is nothing but change in Gibbs free energy. I told you, isn't it? You remember the formula? Del G equals del H minus T del S. I told you, remember in the previous slides. Okay, so the same. If you see the kind of a binding energy, okay. So my plant compound, that is the quercetin, I finalized as a quercetin as the best compound for ovarian cancer for targeting the 10 genes. How I mentioned. So based on the negative maximum energy, when compared to this blue resveratrol and epigenin, I got for FGFR, I got a negative maximum. Similarly, this is the second receptor, and this is the for the third I got, and for fourth I got, and of course fifth also I got here as the best, and sixth also I got as the best. So out of 10, I got 6. So finally, I concluded quercetin is the best dog compound, so which can be used for treating ovarian cancer when compared to epigenin and resveratrol. Even if you check in some literatures, you can find many of them might have used quercetin for treatment of ovarian cancer. So, of course, from this, you can able to conclude that even the computational docking process can give some kind of good results. Okay, but it is not best for few receptors for the 4 receptors. Only it is best for 6 receptors. Of course, even if you check in vitro or in vivo, you may get these kind of results. Okay, I like to show one information. So I told you earlier that I'm working as a professor in Sri Venkateshwara College of Engineering, that is in Sri Perumbudur, near Chennai. Okay, so nearly 30 to 35 kilometers from Chennai. They have excellent hospital facilities for boys and girls, those who are pursuing UG, PG, and PhD. And also, few scholars also completed postdoctoral fellowship in our university. So all these PhD PDF scholars are completed with a stipend. Okay, so they have got already a stipend from CSCR, DBT, DST, etc. Okay, and if you see, if during the initial, if they register PhD, they are going to register PhD in Anna University. They will be getting monthly stipend of rupees 18,000 for the first year. But if they can able to publish a paper in the second year, they may get 20,000 as a monthly stipend. But if the scholar has applied for project proposals and if they get some kind of a stipend from the government organizations, then obviously this will be ceased at that time. And obviously, whatever government fellowships will be getting, that will be continued from the day of sanction order. So obviously, we are promoting even PhD research scholars initially with this stipend. Okay. Currently, 3 scholars. This month, I got one more scholar are pursuing PhD under my supervisorship. Okay. So the scholars are also receiving that kind of a research fellowship. Also, of course, from my side, I am an examiner for PhD scholars in Madras University, VIT University, and also Tech University. I'm also a DC member for um, international students studying in Tech University. Okay, so also mentoring few students, not only in my SVC, and also a few students in other universities, okay, in the form of consultancies, so approved by SVCE. And some advantages of studying MTech program in SVCE. 6 out of 18 students will be getting merit scholarship. That means we refund entire tuition fees for 2 years. In addition, 2 out of remaining 12 students receive CTS scholarship. Again, the refunding will be done for 2 years. Out of remaining 10 students, one can get a merit cum means scholarship and one more get economic means scholarship. It means we are providing much scholarships for the PG students. Okay, so without paying fees, they can study. Okay, so that's what. So nearly 60 to 70% of students get these kind of scholarships. In addition, a separate college budget is allocated for purchase of reagents or chemicals for chemtech students. Okay, this is something about what's happening on research in my institution. Now I take you to the Autodoc Pyrex, virtual screening using Autodoc Pyrex. So Autodoc Pyrex, you remember yesterday was mentioning like this is actually a virtual screening tool. That is, if you want to dock multiple compounds with a receptor, instead of relying on Autodoc Vina, Autodoc 4.0, better go for this kind of Autodoc PYRX. So PY stand for the Python programming, and RX is a kind of a programming we use for docking multiple ligands with the receptor. Actually, when compared to Autodoc software, performing the docking process with multiple library of compounds with a single receptor is easier when compared to the whatever we have acquired upon yesterday Autodoc. Okay, so the process is very simple and even it is efficient. I mean, you'll be getting results faster. Okay. Now I take you to the Autodoc Pyrex. I told you to go to the website, and if you click downloads and you can install that, you can check for the Pyrex software. After installation, how you'll be getting Pyrex. So the Pyrex icon, I better go for Pyrex here. Pyrex. So this is Pyrex. Okay. It looks like the icon looks like an anthem. Okay. So this is it's not a, so this is a Pyrex window. A snake. Okay, a snake is there in a kind of a bulb, something like that. It's a lantern. Okay, this is Python RX programming. Okay, so in this software we can dock multiple ligands with the receptor. So for this, initially I prepare the ligand and receptor. So what ligand I have chosen is guaravinones. Guaravinones or retinoids from Guaravia diffusa. In Tamil, Guaravia diffusa we call it as Mookarattai Keerai. You know, this plant, Verivia diffusa, okay, this plant, Verivia diffusa, is generally used for um, reducing the kidney stones. Even myself, when I faced urinary infection, when I have taken one or two tablets, okay, obviously I found the urinary infection is reduced. So this is very effective on treatment of urinary infection and removing the kidney stones. But in this sample, in our present hands-on experience, I'm going to try with insulin growth factor receptor. Of course, this plant is well to take for diabetic patients. So this can treat somewhat well for the diabetic patients. So what I'm going to do here is I'm going to take some set of Verivnon compounds going to dock with IGF-2. But how I have taken this set of Verivnon compounds, how I have downloaded this file, at least IGF-2, insulin growth factor receptor 2, I can download from the PDB website, isn't it? If you want to download IGF-2, simply go to the RCSP PDB and then you type a keyword. If you know the PDB ID, type the 4-letter PDB ID, that is alphanumeric letter, or simply type IGF-2. You'll be getting not only IGF-2 ligand, but also getting the IGF-2 receptor. Okay, so you can download the protein file from this PDB website. The next is to go how to fetch the uh, ligand file. To fetch the ligand file, better go for NCBA PubChem. In NCBA PubChem, so if you're doing research on diabetes and if you know some plant is good for treatment, you can search some series of compounds, okay, from the PubChem, and we can try to do research. Now, I have taken a plant compound called Verivin. Nearly 14 to 16 compound, I think 14 to 18 compounds, nearly 18. Now we can see 12 compounds are there. Okay, in Verivinone, if I simply type Verivinone, I found 12 results in NCPA PubChem. Okay. Also, a few more Verivinones are there, but the structures are not yet established or not yet posted in NCPA PubChem. You can find in recent literatures, but okay. Now let me at least come across with these 12 results. So when I typed Verivinone, I got 12 results. Okay. Now, if you want to download all the 12 results, you have to simply click download, and instead of coordinate 2D, go for 3D. Check the 3D and then click STF. structured data format. If you click this, then obviously all the 12 variable nodes are downloaded in the form of a single data set, in the form of a STF file. But if I don't want to dock all the 12 compounds, but I want to dock based on the filters, I initially go to the filters. What are the filters? I told you earlier, in NCPA PubChem itself, you can able to see the drug likeness filters. For example, if I want to choose the plant compounds based on the molecular weight, for example, if I want to have molecular weight 300 to 350, okay, 300 to 350, okay, still, or 300 to 330, 6 results are there. Okay, this is one filter. And if you want to reduce further, okay, and if you have one more filter, for example, you want to take only hydrogen bond donor count as 3 and 4. Now I got 5 results. Okay, so from 12 results, now I got after filtering 2 different parameters, drug likeness filters, I got 5 out of 12 compounds are selected. Now simply have to click that X, and then you have to click down. If you see here, now we got 25 results just because we have filtered like that. If you download it, okay, you have to download the 3 format and you have to click STF. Then obviously you will be downloading only the 5 plant compounds in a single data set. Got it? So this is how we can make it out. Okay, so even you can download in desktop somewhere like that. And if you want to open it, okay, again you have to open it using Discovery Studio. I am going to open in Discovery Studio, the latest Discovery Studio 2023. So just once. Now check the structures in STF. Yeah, Discovery Studio is getting loaded. Yes, just 4. I think we have applied third filter also. So totally we got 5. 1, 2, 3, 4, 5. Clear all. But why we are getting only 4? Aware, but some problem is there. Okay, but if you go for downloading, generally download exactly. I better do one thing. I can remove this filter. Now try to download the 6 results. Download now in 3D format, STF. So now as _1. Yes, now I'm going to open it. Open with Discovery Studio 2023. 5. There is some mistake, I don't know. Some one plant compound is obviously. But okay, whatever plant compounds you have downloaded, if you want to see in graphical view, you can see this in the graphical view. Okay, all these compounds are piled together in a single file, isn't it? Only a slight variation with respect to the functional group. If you see the variable nodes, each and every variable node backbone structure is similar. Only the functional groups poses conformations alone is somewhat different with each other. Okay, actually, I've already downloaded all these things in a folder called Pyrex Demo 10. Okay, no, it's not that. It's already there. Okay, do one thing. I take this two a plant compound and a receptor, and I'm going to create one new folder called Pyrex Demo 11. So okay, it's in the desktop, isn't it? So instead of desktop, better go for E drive or F drive, whatever you have. I check whether I have Pyrex Demo 10. Yes, I have a Pyrex Demo 10. I have only 2 files. One is ligand file and another is the receptor file. So first step is to download the ligand STF format. Now this I'm going to open it and show it to you what I have in ligand. In ligand I have 6. So when I downloaded at that time I got this 6. Okay, I don't know this time what's the problem with it. So I got the 6 ligands. Okay, and the ligand the receptor what I'm going to try is IGF-2. Even I can show you the IGF-2 receptor in Discovery Studio 2023. Yes, now this is the IGF-2 receptor. You know why I have taken this protein receptor. This is a smallest protein. This is the smallest protein to work with, so you'll be getting results instantly. So I just taking this protein as a sample file. Now let me start with step one. The step one is to, however, we have done Autodoc. The step one is to check set the path, isn't it? To set the path of input files. So what is the path we had actually? It's not the C users, but presently I had a folder in E drive. Okay, I have a folder in E drive. So I close one window. So to fetch the path, you simply click here, try to copy the path. But ensure whatever folder name you're going to create, there should not be any space. What I did is I used pyrex underscore demo 10. Even don't use pyrx underscore demo spacebar 10. That is also wrong. Pyrx spacebar demo 10 is also wrong. You should not have any space. Okay, if you have two words, better use underscore to match both the words. Now my workspace is E underscore the particular folder name. Simply click OK. So I am getting a default parameter available CPUs as 19 because in my laptop I have number of cores and threads are more. Okay, in i7 laptop I have more cores and threads, so obviously it generated around 19 CPUs. But if you're using a Core i3, it may give only 3 CPUs. Even i5, I think 3 to 5, not more than that. So better use a high configuration, and that too, even when I tried with i5, I can't able to dock not more than 50 compounds at a time. If you download 1,000 compounds and if you try to dock, obviously your system can show only 50 results, up to 50 results, and remaining results you find even your laptop got hanged for a long. Then you have to uh, shut down improperly, and then you have to restart and you have to work once again. Okay, so based on the processing power of your laptops or desktops, whatever you use, you have to take a data set according. In drug discovery hackathon, I have downloaded nearly 1,152 plant compounds and docked with the spike glycoprotein. But how I have completed that 1,152 plant compounds? You have any idea? I splitted that 1,152 plant compounds into various data sets. Okay, various data set files, and each and every data set file is not containing more than 50. So just like that, I split it into my various files and then just started doing all that. Okay, let me go to the step two. The step two is something related to how to load the macromolecule. To load the macromolecule, click load molecule. Okay, but defaultly, it's not taking you to that path. You have to simply locate the path. The path what we have selected is the pyrex demo 10. In the pyrex demo 10, what molecule we have to load? The macromolecule to be loaded is IGF2. So simply click this IGF2 and click open. Now, if you click this protein molecule gets loaded in this window. But see here, whatever IGF2 molecule I have downloaded is a hypothetical protein downloaded from the PDB database. So when I got nearly 20 models for it. It's a model protein, so I got 20 models. There is no need to use all these 20 models. The first model might be a best model during the generation. So what I'm going to do here is I'm going to remove the remaining 19. I restore only model 1. I'm going to remove the remaining 19. So I got only one model at present. If you click this, if you expand this, you can find all the amino acids in that one model. Okay. If you want to check how many amino acids are there, nearly 67 amino acids are there. In AGF2, we have 67 amino acids. Now, the next step is to click here. Okay, right click there or go to Autodoc, and you have to make macromolecule. You know the significance of this step? I told you the Autodoc generally uses PDBQT files. It uses only PDBQT files. It won't use PDB files. Even if you download PDB files from the PDB database or MMDB database, you have to convert it as Autodoc PDBQT files. Okay. So all the Autodoc uses the same kind of principles. So if you click this Autodoc and make macromolecule, So obviously this macromolecule is converted as .pdbqt. How it is converted as pdbqt. For to know whether it's created or not, click here Autodoc. Instead of molecules, I'm going to the Autodoc here, and here you have plus, isn't it? Expand it. If expand it, you can see this. My protein molecule is now going to save as pdbqt in that step. Okay, you got it. So right click Autodoc macromolecule. That's it. Now the next step is to even convert the ligand, whatever you downloaded. Whatever ligand we have downloaded from the NCBI PubChem is in what format? Can someone tell me whatever ligand data set I downloaded from PubChem is in what file format? What file format did we download the ligand? Remember, I downloaded nearly 5 to 6 query nodes. What is the format of that file? Good. Already one answer, Anushka. STF is the correct answer. Structure data format, isn't it? Okay. Now I take you to the Pyrex window. So even the STF format file has to be converted to PDBQT and not PDB. Okay. So for that, what is the step we have to make? For this, you have to go to the Autodoc wizard. No, Autodoc wizard. Go to the Vina wizard. Can go for you can go for Open Babel. The next step is to go for Open Babel. If you click, because we didn't convert the ligand as PDBQT, isn't it? So first we have to use a software called Open Babel or even Babelwin in Babelwin is a software which it can able to work without the internet. But Open Babel requires internet. Okay. So actually we are going to use a Open Babel software. But the Open Babel is built in the Pyrex. So there is no need even to have internet to work on Open Babel. But if you want to convert the STF into a PDB format, you can convert it in even in Chrome using Open Babel software. Now the next step after selecting the Open Babel is to click this green icon. You have insert new item. Okay, insert new item in this bottom. You have to click this. So you have to select the ligand. So the ligand is present in Pyrex demo 10, isn't it? So nodes are the ligands. I actually am going to select 10 nodes. STF format. I got all these. I mean, 6. Only 6 I downloaded, isn't it? So I got all the 6 in the STF format. So now what I'm going to do here is first click only one and right click. You get minimize selected. So the what I'm going to do here is for, initially I'm going to calculate energy by energy minimization methods. In molecular modeling, we have one concept called energy minimization. We generally use steepest descent or conjugate gradient, or it's a combination. Okay. So generally we use steepest descent or conjugate gradient energy minimization methods for making a ligand more stable during binding with the receptor. Even same energy minimization we can also carry out with the proteins. So to make protein folding more stable, we generally go for calculating energy initially. And after energy minimization, we can also calculate the energy. You can find the energy after minimization, you find somewhat negative maximum when compared to before energy minimization of both ligand and receptor. But now in this step, I'm going to, instead of doing the energy minimization over a single ligand, I'm going to use all the 6 ligands for docking, isn't it? So instead of clicking minimize selected, click minimize all. If I click minimize all, the energy minimization has happened with all the 6 ligands. The energy is shown here now. Now, the next step is to right click and convert all to Autodoc ligand. So now what is going to happen? All the ligands are converted as .pdbqt files, all the 6 ligands. So the process has happened now. All the 6 ligands are converted as PDBQT. Okay, so if you want to check, okay, We can go with the winner wizard. Okay, can check yes. We have IGF2. How about ligands? Yes, if I double click the ligands, I can also find all the 6 ligands are converted as PDBQT. You remember what I did initially? Go on to the open Babel, click this insert new item, opened all the 6 ligands, right click the first one, minimize all, and next convert all to Autodoc ligand PDBQT. So all the ligands are converted as PDBQT. Obviously, if you click it, it expands and it shows all the PDBQT ligands. The next step is to go Vina Wizard. If you click the Vina Wizard, you know what results I got. What input parameters I have selected. Here, no ligand got selected at present, but the protein molecule IGF2 got selected. If you want to dock all the 6 ligands with the protein receptor, then you have to select all the 6 ligands. So if you want to, if you select, if you click this, and if you click shift and you select all the 6 ligands, then you can find a parameter 6 ligands selected. Okay. If only if you select all the 6 ligands, and if only if you select the protein receptor, okay, then only you can go for docking. So the next step is to click forward. If I click forward, I am getting a kind of a grid box. Okay. In the grid box, you have to click this cube. If you click this cube, obviously you can find the protein molecule is present in 3 dimensions in the grid box, isn't it? Do you remember this step in Autodoc on yesterday? Similar kind of a steps we are also doing it here. Okay. So to if and if you enclose the entire protein in a grid box, then only the binding sites of both hydrophilic and hydrophobic nature can be determined by flood fill algorithm or any other related algorithms. You know, in this visualizers, okay, any visualizers, if you are using a liquid kind of a sample, then obviously water is the control. If you are using a gaseous molecules packed in the grid, then obviously air is your control. AIR is your control. Likewise, so each and everything, okay, whatever they develop, even the protein, each and every atom of an amino acid of a protein is also a predetermined one. Generally, wherever you find red color, the red color atoms are oxygen atoms. Wherever you can find white color, the white color are hydrogen atoms. Okay, that is standard. Okay, fit in all these softwares, not only in the visualizers but also in the docking come visualizers. Okay, now what I'm going to do here is I'm going to fit the entire protein in this cubical box, the cubical grid box. Okay, I've enclosed the entire protein in this cubical grid box. So after fitting this protein, okay, after this fitting this protein, if you have a large grid box, then obviously it takes much time for docking. So I just fit out accordingly. Now click forward. If I click forward, obviously defaultly it going to run the auto grid, and also running the Autodoc happens after running the auto grid. A single button forward is doing both the steps. Okay, whatever we completed on yesterday. See, 3 ligand molecules are already docked with the protein receptor. Fourth is also completed. Remaining 2 more is left out. So what is happening? The status is mentioned here. Nearly 20 CPUs are detected. So just because the processing power is more, I'm getting somewhat faster. Okay, the results we have got it. So this is the final result. This is the final result. And if you see this first molecule, the binding affinity, what we got here is minus 7.2 for the first conformer. Okay, we have results for each and every ligand. We have for each and every ligand we have generated totally 9 poses. Only 9 poses are generated. 0 to 8. 0, 1, 2, 3. So 9 base poses are shown here for the first ligand. And similarly, for the second ligand, we got 0 to 9 poses. Okay. So obviously, whatever shown in 0th pose is the best conformer. If you ask me about the first ligand, what we dock, the best conformation we got is minus 7.2 kilocalorie per mole. But in the second ligand, the best conformer energy what we got is minus 7.1 kilocalorie per mole. So the second ligand is not best when compared to ligand 1. That's the conclusion. Now, still we go for the third ligand because we have used totally 6 ligands, isn't it? The third ligand is minus 6.5, which is somewhat more when compared to minus 7.1 and 7.2. This is not significant when compared to ligand 1 and ligand 2. So this is how we can infer the result for the third ligand. Go for the fourth, minus 7.2, significant. So ligand 1 and ligand 4 are somewhat significant when compared to the others. And now we can go for the next. Minus 6.9 is not significant because it's a maximum value. Minus 6.8 is also not significant for the sixth ligand. So finally, I conclude ligand 1 and ligand 4 are the best. So if you want to download the results, even I can download the results, okay, by saving this as comma separated value file. Whatever Excel sheet, whatever data you have, all these data are stored in this Excel sheet. You can just type the name and you can open later it in the Excel to find. So if you want to see this, okay, I just show it in the desktop. Okay, so this is the result of Varivinons with IGF2 complex, isn't it? So this is the such a kind of CSV file. I'm just going to save this. So whatever I saved, I'm going to open it in the desktop now. So in the desktop, I'm going to open this. So whatever information I got, not only the binding affinity, the root mean square deviation, you see here, the root mean square for the zeroth pose, is 0. But if you see for the next poses, you find a deviation. If you find a deviation, that is not best. Okay, so you can conclude that, of course, for the first ligand, this is the best pose. And what is the PDB ID we got? 134940642. I just copied that alone. To know what is the ligand name, okay, I just going to type that PubChem ID here. If I type the PubChem ID, I got this as, what is the name of this compound? I just going to click inside and going to check it. It is the name of a kind of a Beryllium derivative. Okay. So there is no such Beryllium specific name, but it is a kind of a derivative, a Beryllium derivative. Then I can go for compound 4 and I can check it. So 0, 3, 4. Oh, yeah, this is compound 4. I'm going to check the name of this compound 4, which is also showing minus 7.2. So I just copy it and going to paste it here. Okay, I'm going to check what is the fourth compound. The fourth compound is also a kind of a rutinoid. Okay, and for this PubChem is mentioned here. So it's a kind of a rutinoid. So this is the name, IUPAC name. Okay, so these two plant compounds are docking well with the IGF2 receptor. So this is how we can control. You know, in autodock Pyrex, we can get only the energy values. But if you want to get hydrogen bond interactions, and if you want to get the other data, whatever I showed earlier, you can't able to get this data in Pyrex. Then what is the best software? So actually, this software is used for just screening the best docking ligands. If you see out of 6 ligands, only 2 ligands are docking well. So the next step is to use these 2 ligands separately for docking process with IGF2 receptor using autodock. First, you have to dock with ligand 1. Then you have to dock with ligand 4 with the same IGF2 receptor in autodock, like how I performed on yesterday. Okay, this is how we can take steps. Okay, now let me come across with a complex file we have generated on yesterday. Do you remember the complex file we have generated on yesterday? So this is the complex file we have got when we docked indinavir with HIV protease. Inavir is the general drug used for protease. It's an inhibitor for HIV protease. So I docked it using autodock, isn't it? So this is the protein. And now, if you want to see the kind of 2D interactions, simply click here, receptor ligand interactions, click show 2D diagram. So this is the 2D diagram, isn't it? So in this 2D diagram, if you want to check the bond length, right click here, display style, okay, and even you can able to find the distance, the bond length. Okay, if you see here the hydrogen bonds, you have both classical hydrogen bonds and non-classical hydrogen bonds shown here. And other than that, for this interaction, you also find hydrophobic interactions like alkyl, hydrophobic, mixed pi-alkyl. Okay, so whatever interactions you have, this is automatically active. This showed highlighted and automatically active. And now simply you have to click here, show distance, apply, and OK. See here. Now you also got the bond length. The bond length from here to here, the glycine A4 to A2 here is 5.04. And similarly, isoleucine A54 to the ligand is 5.23. So this is how we can also get the bond length data. This is how we can take it and we can mention in the data table. That's what I did in the journal papers. Later, if you want to see the hydrogen bond contacting between the ligand and receptor, that also I showed, isn't it? So instead of simply going with the 2D interaction, even we can get some information in the 3D interaction. Further, the first step is to click view hierarchy. If you click view hierarchy, you get a chain with amino acids. Okay, also your ligand is shown here. This is the ligand, intinavir. And if you select this, this is the HIV protease. Okay, what is the first step we have to do for getting the detailed hydrogen bond interactions information? For this, the first step is click scripts, selection, select ligands. Now the ligand alone is got selected. Next, scripts, selection, select atoms within 4 angstrom. If you want to select the 4 angstrom binding site amino acids nearer to the ligand, you are using this step. One is selection, select ligands. The next step is selection, select atoms within 4 angstrom. The next step is to click ligand interactions, show ligand binding site atoms. Scripts ligand interactions and show ligand binding site atoms. See here, a new hydrogen bond monitor is generated. Now, after clicking this third step, click here. If you see here, my ligand is MK1902, that is intinavir. You have to check where the intinavir is going to bind. The intinavir is binding with the intinavir atom itself. So, actually, if you see here, we don't have hydrogen bond interactions between intinavir and HIV protease. That's the conclusion. If you see here, the amino acid atom of one protein is contacting with other amino acid atom of the same protein. So one is invalid, second is also invalid, third is also invalid. The fourth is ligand with the ligand is going to share a hydrogen bond atom. So there is no such proper hydrogen bond interaction between the ligand and the receptor. So this is the conclusion. Okay, if you see some other kind of a demos, I think I might have some other demos. If you see some other complexes, if you dock it well, okay, you can also find hydrogen bond interactions. But in our case, we don't have hydrogen bond interactions. Okay. Okay, I think I have completed my explanation. Any doubts you have, participants, you can ask me. So today I have completed how to go for docking analysis, and even the validation can be done through one more website called PDB sum. If a PDB structure is known, and if you want to determine the binding sites, the binding sites can be determined directly even from UniProt and PDB sum. For example, if you want to find the binding sites of the IGF2, okay, simply type IGF2. Okay, you'll be getting a receptors, and if you are working on human, check for the human one. So click this UniProt ID. Okay, I've gone for the IGF2 human. Go for the binding sites by scrolling down. See here, this is the binding site. R48, arginine 48, arginine 58, arginine 61. All arginine molecules are actually shown as a binding sites. Got it? So even if you dock the hydrogen, if you get hydrogen bond interactions with any of these four atoms, you can tell your drug is well uh, binding with the IGF2. So it can be used for treatment. So Indinavir can be used, but we don't know how much efficacy the HIV protease. So that's the conclusion. Got it. So however, I got the binding sites for the Uniprot. Even if you know the protein PDB ID, you just have to simply either you can find from this or you can type from the AlphaFold model. Okay, just I typed one PDB ID and I am checking the binding sites from inside. Okay, you have to click protein inside. You see the protein here. We don't we have only one chain, but for ligands, if you click, yeah, you have one ligand. The ligand is binding with a few amino acids. Okay, so the ligand binding site amino acids are shown here. So even the binding sites can also be seen here. Okay, in the PDBSum. Either you can go to the UniProt or you can go to the PDBSum for determining the binding sites. So the validation of docking can be done like this. So how to go for docking analysis? The docking analysis through Discovery Studio, validation through UniProt or PDBSum, and virtual screening through the Autodoc Pyrex. Okay, I've completed my session now. Any doubts you can, I think no one has typed anything in the chat box. If you have anything, you can type it. Or else, thank you for your patience. And also we have sessions on tomorrow and day after tomorrow at 7 o'clock. So join sharply. If you weren't able to join the sessions, watch the recorded sessions. Okay. Thank you. Have a great day. Have a great weekend. Bye. Good night.